So this lecture is going to be about some ways that we can use sequencing to understand organization of chromatin in the nucleus. And so the picture here is actually a reconstruction. These are mouse chromosomes reconstructed from 3D interaction data, and so you can think of this as it's an ensemble. But basically what we have is we're coloring these with active transcription in red and lamin in green, and so you can already see starting to capture some of the features of the nucleus. These are just pretty pictures, but it's actually possible to start to reconstruct these sorts of things. So let's start really basic though. So, for me, the problem I've been interested in forever is this one. We know we've talked a ton about cis-regulatory modules and promoters. Ross defined all of these things operationally, but there's always this question of what's the mechanism by which a cis-regulatory module, whether it's a kilobase, 10 kilobases, or 300 kilobases away from its promoter, actually influences the rate of transcription. And so a particular class of regulatory elements and corresponding transcription factors that I've been interested in for a long time are these insulator proteins. And so Ross asked me if I was going to talk about insulators, so I decided I would give you a little bit of background. So we're going to go way back. So this is yellow in Drosophila, and so you've got this interesting phenomenon. You have 5 different tissue-specific enhancers here, and so these are some of the tissues in which each of these enhancers influences expression. And there's a particular mutant from this insertion of a retrotransposon called gypsy. And in this mutant, you would see that yellow expression was normal in these various regions, but not in wing blade and not in body. And so what the other interesting thing, though, was that there's a corresponding rescue of this when you see a mutation upstream of a protein called suppressor of hairy wing. And so there's a lot of biochemistry that need to happen in here, but eventually the binding site for this protein was figured out, and it turns out this gypsy retrotransposon contains a cluster of, I believe, 12 binding sites for suppressor of hairy wing. And so the inference here is that suppressor of hairy wing binding to the site is actually somehow blocking these 2 enhancers from influencing the corresponding transcriptional unit. And so you can, if you get down to just that minimal binding site now, and we're inching down from this retrotransposon to trying to get the minimum element that's effective and insert it at various locations between all of these enhancers, you can see that regardless of orientation, right? So regardless of which orientation you put the binding site in, and regardless of its position relative to the transcription start site, it consistently blocks the enhancers that are away from the location where the element was inserted. And so this is that canonical enhancer binding, enhancer blocking activity that Ross was talking about. Now, one other thing just to mention is that the enhancer blocking really has to be only for enhancers that are actually blocked, if that makes sense. So it's important that other enhancers which are still located near, without the insulator in between the transcription start site and the gene are still active. And so this isn't a regional effect, right? It's a fairly punctate effect that only blocks at that position. And so as of now, we know of on the order of 6 insulator proteins, and so suppressor of hairy wing was the one that was just talked about. These are 4 that occur in Drosophila. There may be one other, depending on how you define these things. They all involve this CP190 protein, and most of them require this mod cofactor. The only one of these that has a homolog outside of insects is CTCF. And so CTCF is basically the only insulator in, for example, mammalian genomes. Now, there is TF3C, which is the only insulator in yeast, and there's some evidence that there may be some TF3C insulators in other species. But for the most part, this is the key insulator protein. Now, the other interesting thing, if we go back to yellow, this was a really kind of cool paper in 2001 that took constructs with yellow and white and the suppressor of harrowing protein, but also looked at a couple of other cases. So you've got your standard, you've got, this is an enhancer for white, or rather wing and body, and then we have the suppressor of harrowing sites. And so what you see here is this is whether yellow is activated or not. And so if you have one suppressor of harrowing cluster, then you stop activation. But if you have two, this is actually restored. And so you can see other cases like that. Right, and so this is a very interesting case where actually pairing the site restores activity. And similarly, let's see, what was I looking for here? Right, so even if the pairing is surrounding a gene like this case, right? So here, where we have suppressor of harrowing site on either side of this yellow gene, but we have both of the relevant enhancers upstream, yellow is off and white is on. And so you can, the model that's kind of hypothesized would be something like this, right? So that alone, one of these proteins is actually just having this enhancer blocking activity. We don't, how is a relevant question, but the claim would be, okay, these things somehow can pair up and actually even increase the rate of transcription at this locus, because it's not acting as a blocker anymore, but it's actually moving the enhancer potentially closer to its promoter region, and perhaps even 3 or even 4 of these things can pair together to create these large enhancer bodies. Now, is there any other evidence for this? Well, you can see it. So this is a case where we have, in wild type, we have a loop, and what's down here are copies of that same gypsy insulator sequence, and so they've got the same suppressor of harrowing binding sites, and so at the base of the loop you see interaction. And out here you don't. And so if you actually have a mutant where you put another gypsy insulator sequence, right, that 700 base pair sequence, inside the middle of this loop, you form a complex and actually create 2 new loops. Now, we could argue about what you're actually seeing there, but there's some evidence that corresponds with this image. And so this kind of gives us a cartoony model of how these things might work. You can use these proteins to bring together certain regions of the genome, and in doing so, you're partitioning transcriptional units, so groups of genes that are going to be co-regulated in particular cell types. Okay, so arguably the term insulator protein is actually not a very accurate way to talk about these, and so I will propose to you that architectural protein might be a more legitimate way to talk about it. All right. So back to the question of how do we form these links? Well, there's a couple of ways that I already talked about earlier, right? So one was the idea that we can use EQTL type mapping to make these kinds of links. And so here's one of these examples where you have variants that are associated with changes in expression, and so this, particularly if they're associated with other regulatory element features, might be a way that you could make these links. We can link things through correlated activity, right? This is basically saying, okay, if I have enough cell types, and you need a decent number of cell types to have any statistical power here, but this is something like 8 encode cell types, and we've got expression at the top, and then we've got K4 mono and K27 acetylation. So what you're seeing in this cell type, you have expression. You also have fairly strong K4 monomethylation. You'll see you also have some K4 monomethylation in other cell types, but then here you have a fairly strong signal for K27 acetylation. And so perhaps these are, poised in other cell types, but through looking at these correlations, you can make a link between the putative regulatory element that's sitting out here and this gene. So that's all good, but another way that is a bit more direct perhaps that we can try and get a high throughput measurement of this is to try and create a sequencing problem that gives us some kind of measure of actual 3D proximity in the nucleus. And so the enabling technique for all of these approaches is something that was published over 10 years ago by Job Decker called capturing chromosome conformation or chromosome conformation capture. The technique is referred to as 3C. The idea is, okay, so we have some genomic DNA drawn as a loop here, but perhaps there's much more complicated stuff going on out here. But the important thing is somewhere there's an interaction complex of two regions of the genome that are linearly distant but spatially proximal. And there's some DNA and some protein involved here. So the first thing we can do is cross-link this. There's different ways, even at this step, you could think about doing things. The more recent protocols, which seem to have the best noise properties, are able to do this cross-linking and the subsequent steps in the nucleus. And so those are probably a better way to do this, but nonetheless, what you're going to get is cross-linking across the entire genome where you have proximities. You're going to go in and you're going to cut with a restriction enzyme. There's an alternative protocol that would do DNase digestion here for fragmentation, but most of these protocols use restriction fragments, and there's good questions about what particular restriction enzyme you want to use. That's going to determine the length of your fragments, which is going to determine various aspects of resolution. But typically, this is going to be a 6 base pair or 4 base pair cutter, depending on what you're trying to achieve. And so once you've cut, you now have 2 chunks of DNA, which I've colored now white and gray, which are from different regions of the genome. And so you can ligate those, and if done well, most of the time you're going to get a ligation between 2 fragments. You can purify, and now we get these ligation products. And this is what makes the 3C library. And so you can now go into the 3C library and quantify these junctions. So this is where the use of the restriction enzymes is important, because you know particular expected interaction points at the junction of 2 cut sites, and so you can assay a particular set of interactions that you're interested in. So far, so good. So that allows us to target a few interactions. The question is, can we go genome-wide? And so in 2009, this pretty transformational paper was published, which took this technique and turned it into a genome-wide high-throughput assay. The key to this is actually modifying the 3C protocol at the point where we have, before we do the ligation, to do end filling with biotinylated nucleotides. And so now our products, after we purify them, have this biotin at the junction point. The reason we need that is because we want to do high throughput sequencing, we need to get these products shorter. And so we can shear them, and then using, by selecting out these fragments, we can throw away all the pieces that don't represent a junction. And then now we have we size select this. Now we have fragments that are reasonably sized to put onto an Illumina sequencer, and so we can do paired end sequencing. And what you're going to get is basically paired end sequencing reads, where one read will come from an end here, the other read will come from an end here. The fact that they're paired tells me that the whatever restriction fragments they each map to were in spatial proximity. And so the efficiency of this is fairly variable in terms of the number of useful reads you get out. But if in this particular first experiment, this was only 8.4 million read pairs. And so what you're seeing here now is a heat map in which the rows and the columns are positions in the genome. And in this case, this is an arm of chromosome 14 against itself, right? So on the diagonal, those are, of course, things that are interacting either with themselves or very nearby. And then off diagonal, this is a level of interaction between this region and this region, right? So each place where you see, each value here is the amount in some quantity related to the amount of interaction between two regions. So do these actually mean anything? Well, frankly, at this resolution, it's hard to say anything really strong, but they did a couple of things in this paper. So they start with just the observed raw data. They came up with a polymer model for what you would expect it to look like just if things were interacting with each other by chance, and generated some expectation, come up with an observed over expected, and did a PCA basically analysis on this and looked at the first eigenvalue. And so it actually segregates out somewhat, where basically you have things are either strongly positive here, strongly negative, suggesting an overall organization into two compartments. And if you go back and take regions that are in either strongly in one or strongly in the other component, you can verify this with FISH. And so that's one of the first observations here, is that the nucleus is in fact organized into two compartments. The other very, very pretty observation from this paper was that if you look at the distribution of distances, so all it's saying is taking those interactions, for every single interaction you're looking at how far apart on the linear genome they are, and you get a distribution from that. And it has kind of a power law distribution, so this log distribution. And in this kind of middle region here, it's got a slope of approximately 1. And then if you simulate from various potential models for the genome based on these polymer models, you'll find that just an equilibrium model, which is what, the conformation of the polymer where it's got the lowest energy, is this all knotty structure here, is not consistent with the observed distances, and instead this fractal globule is. And so what came out of this was a suggestion that the genome might be organized as a fractal globule. It's important to recognize that these pictures have nothing to do with the data, right? This is not a picture of any actual chromatin. It's a hypothetical model that is consistent with a single number that is derived from the chromatin. So this is totally hypothetical. But nonetheless, this is a potential model for how the nucleus might be organized, which is at the top level you have chromosome territories, and then below that you have kind of a succession of knots in this fractal globule state. And this is appealing because what it means, this is very hard to untangle. Right? It's at equilibrium, but that's when it gets to equilibrium, it's tied itself up completely. This is pretty easy to unfold. So if you want to change the chromatin state, because the nature of something that's fractal, right? It's nested and nested and nested, it makes it easy to unwind. Cool.
What is the distance the distance between any two?
Every two, right? So you take your read pair and you just say how far along the linear genome is where one end maps the other end. That gives you a distance. And that is the distribution of those distances. So you have, as you'd expect, many, many things that have distance that is very small, and a few things that have distance that's very large.
So I'm just thinking about the polymer model, about how that would be a very hard thing to untangle. Like, as a considered model in this, it seems like you can throw that away pretty easily, thinking about all the remodeling that needs to happen, not only in gene regulation, but just in cell division.
I agree.
Okay.
Yes.
So.
Yes. So, it's one of these things that once you think about it, it's like, well, of course, right? And how long would it take to actually come into this conformation? I don't know in their simulations how hard it is to get this, but it would be way, way too long to be practical. But from a biophysical perspective, I'm sure they were like, oh, of course it's going to be in the lowest energy state. Why wouldn't it be? Right? Other thoughts? Okay, so zoom forward. Really what's happening now is just sequencing is getting cheaper and higher throughput, and so we can start to do these experiments in much more detail. And so Jesse Dixon and Bing Ren, 2012, published this paper where instead of doing 8 million reads, they were doing on the order of 2 billion sequenced reads across a couple of cell types. So they looked at human and mouse ES cells and human fibroblasts and mouse cortex. Like I said, 1.7 billion read pairs. They map them back to the genome just like I said. They bin the genome into 40 kb bins or 20 kb bins and just then count for every pair of bins how many things are aligning in how many reads are in one side and the other. And so you get something that looks like this, much more resolution, right? So this is, yeah, there's scale on here. Okay, so this is about 4 megabases, I think. And you've got the same heat map. All that's been done is this is the diagonal of the heat map. It's been rotated 45 degrees and pulled down. And so this structure very obviously emerges. And what they did is they came up with a score that basically says for every region, look at the ratio of things that are mapping upstream versus downstream more. And so what it brings out is at the boundary, you see changes in the score. And this is actually, they just ran an HMM over that and get out segmentation. And this is the segmentation. And so these things have come to be called topologically associating domains, or TADs. And in this paper, they were said to have a mean size of 800 kilobases, and this is a reasonable definition. At the same time, like I told you, the way they determined these does require a certain thresholding, and it also is dependent on the resolution of your sequencing. So it's not exactly clear what the size of something that's called a topologically associating domain is. But they're interesting. They seem to be a fairly robust feature of the genome. Right? So basically, one way of thinking about this is at the boundary between a domain, you have DNA that's self-interacting upstream, and downstream it's self-interacting, but you have, for some reason, greatly reduced interaction between the things on either side of that domain. And if you look, so this is ES in mouse versus cortex, there's quite a bit of overlap. Yeah. So these are said to be conserved between cell types. I will ask you to read the numbers here because there's almost as many that are ES cell specific as overlapping. And so, they are relatively stable features, but they do differ somewhat between cell types. Down here in human, this is human ES, IMR 90. Here you're seeing a bit more consistency, and so they do appear to be, in aggregate, stable features. Okay? All right. So from there, let's ask whether we can resolve smaller structures. So Hi-C definitely has some problems. We'll go, at the end, I'll show you some really high resolution Hi-C, but it's because of the nature of these, the background distance dependence, even with a lot of sequencing, it's hard to discriminate from background, and it's hard to get things that are at really long ranges. And so we'd like to come up with some kind of enrichment approach that allows us to target regions of the genome that we want to focus on. So nowadays, there's probably 10 of these. There's at least 3 distinct protocols using some kind of solution capture to enrich for regions. There's various approaches for combining this with CHIP. So if you wanted to enrich for interactions involving a particular protein, you can use ChIP-PET or chip loop or a variety of other techniques. I'm going to talk about one that uses an amplification-based enrichment, and that's chromosome conformation capture carbon copy, or 5C. But a lot of the analysis issues I'm going to talk about apply to all of these technologies. And so here, this is how this works. Is basically we have our 3C library, just like before. But what we do is we actually design probes for all of the regions that we want to interrogate. So what we have here is basically a probe that's complementary to one side of an interaction we want to interrogate, another probe that's complementary to the other, and then universal primers. And so this uses something called ligation-mediated amplification. So what's going to happen is if in your 3C library you have lots of this product, then this is going to amplify. And so what you get is not your products, but actually these amplified primer pairs, and the amount of them you're going to have is going to be proportional to the amount of interaction that was in the 3C library. Proportional, of course, but exponentially biased due to PCR. And so what you get, you do this for many primers simultaneously, right? So I would go in and I would design a few thousand of these primers, and that would allow me to interrogate all pairs of interactions between these regions. And so basically you have all of these different little PCR products. And so at the end, I have sequences that look like this. And so the relative abundance of these tells me something about frequencies of interactions. I'm going to give you one example of this. We're very interested in gene regulation below a megabase, and so one way you could do a capture experiment is to alternate across these regions. And so here's what one of these designs might look like. So this is about 1.5 megabases, and for this to work, you can only interrogate every other fragment because the primers have to be oriented at opposite ends of the cut fragments. And so basically what you can get is a tiling of an entire region where you interrogate 50% of the possible interactions. Okay, so what does the data for any of these look like? I'm going to show you 5C because it's a bit easier to look at, but this is absolutely the same for high C, or for other where you've done other kinds of capture that aren't tiled, you're going to see lots of empty regions. So it's easier to look at these high density plots. But this is showing, in the case of 5C, our forward probes and our reverse probes. And we have a single region showing log counts. And if we zoom out a little, see something like this. And so it's fairly obvious that there's a lot of bias in these experiments. The striping is telling you that the overall level you observe for a given primer has, right, there's an enormous influence of which primer you are on what you're observing rather than the actual pair of interactions. So that's why you see all of these stripes. You would expect something more like this. You expect symmetry, right? So you would expect that since this region is next to this region, that these two things, which are right next to each other on the genome, would look similar. And yet instead, we see these strong either low points or high points. If you just plot these out in box plots, this is again on a log scale, we see several orders of magnitude of variation in the signal you see for a given primer. And so this is very, very highly biased. One question you might ask is, can we correct for this experimentally? And so for 3C experiments, kind of a classic way of doing this would be to try and use a control library from perhaps a set of backs, do the experiment on that, and then whatever variation you see should be not associated with biology. And you can do this for 5C at least, and get something like this that has substantial bias. The problem is that it turns out to be extremely difficult to, because of the amount of amplification that goes in here, it's really, really difficult to get this normalized well. And so we see just as much variation within the backs, but also between the backs. In this case, this has actually been optimized multiple times to try and get the concentrations the same. And for something like high C, this is pretty impossible. You could choose a set of representative interactions to try and normalize for, but you're not going to be able to do the whole thing. And we've looked at this a lot. The other issue is it's actually not really clear that the back data estimate the true effect well, because whatever's going on with structure. So the intuition, though, is that although we have only one observation per interaction, we have many observations for a given probe or restriction fragment, and thus we can we can use all of those to model a probe-specific effect. And so these are the approaches. We've developed some of these approaches. There's a lot of approaches there's several programs and just a couple of approaches actually for modeling this. And so the first is this idea of explicit modeling of biases. And so this was the first idea to come along, is say, okay, is it GC content? Is it length? And those are very strong, and you can model those in a regression. And so this is a reasonable way to normalize, but in my opinion, it has room for improvement. So there's a couple of these more per fragment or fragment n kind of corrections that rely on that idea that you're expecting symmetry again. So matrix balancing might be one that's fairly easy to think about. If you're expecting symmetry, what if you just have a correction factor and look at the matrix and ask how much do I need to change that correction factor to make it more similar, to make it more symmetrical, right? So if I increase every value in a row and a column by a certain amount, or decrease it by a certain amount until I get closer to symmetry. And then you just keep doing that iteratively and iteratively and iteratively until you come up with a set of scaling factors for every row and column that make the matrix as balanced as possible. That's a very intuitive idea. It's also kind of ad hoc, maybe less principled. We can also think about probabilistic models where we are actually trying to put this data into a reasonable distribution and then learn normalization factors using some kind of maximum likelihood approach. And so this is just a sketch of the kind of thing that we do in these probabilistic models. And so you have the expected interaction count, in this case log interaction count between two regions. Well, let's say we have a probe effect for one, a probe effect for another, and the distance effect, which is just how much interaction you expect based on how far apart they are linearly on the genome. And given an expectation of a distribution, we can learn all of these F correction parameters. And so in one way we can do this is using a gradient descent approach. If we do, we can get something like this. And so this is our original raw data. This is probe-corrected data, and so the structures start to emerge, the biases are reduced. The other thing we can do when we have a full probabilistic model is not just say, I want to correct this. I can say, I actually want to ask for every region, where does it sit in terms of this probability distribution? So now I have something I can actually interpret that is independent of distance, which I think is very, very important, right? So I can look at two regions and say, which is interacting more or less without having to think about how far apart they are in the genome. We've taken out that confounding factor. And so this is what you really need if you want to start actually making significance calls about these interactions. Let me give you one example from our own work that kind of illustrates how the high resolution stuff looks. So this is some work we did in cell lines. So basically differentiating mouse ES cells down to neural progenitors. And we use this, this megabase tiling approach on a few key loci. Now, the interesting thing is at that resolution, we actually find a fairly different looking structure. And we find that the small scale structures, the sub-megabase structures, are quite a bit more dynamic between these cell types than the large scale TAD structures. So the overlap here is smaller. Each of these has a fairly significant number of structures that are unique to them. This is just an illustration of the regions showing different interactions. If we just look at this in the heat maps for an illustrative example, so this is in the ES cells, and we can see we've got a domain here that's highly compact in our ES cells, and then down to our MPCs. Now we've formed different domains, and this is opened up, and this happens to be where one of these key genes is. And so this is a fairly visible and dramatic change between two cell types, more than you could see in the at the high level structures. And so another interesting thing here perhaps is that we see a few proteins that are involved in these. And so CTCF certainly is, but it's involved at these fairly long loops on the order of megabases, back to kind of the TAD level structure. It's also involved in medium loops, but when it is, it tends to be associated with cohesin. Actually, at these loops, it's not significantly associated with cohesion. And then at the smaller loops, we find this mediator cohesion structure that's very depleted at the long range and very enriched at the short range. So I would argue that there's definitely different structural characteristics at different length scales, and especially in terms of cell type specificity of structure. Okay, so a little more informatics. So one of the things that we probably want to do is actually call these boundaries. So this is how that Dixon paper, I mentioned this directionality index before, and so the idea is basically that we look at reads mapping from a given point in a 40 kb bit upstream and downstream. From a given bin 2 megabases upstream versus 2 megabases downstream, right? So you're basically looking at everything that's in this bin, counting the number that map in the 2 megabases upstream and downstream and taking a ratio. And so that's going to give you this index, and when it hits 0, you're basically equally upstream and downstream. And so that's how they're kind of defining these boundary points, is when you have no mapping bias upstream or downstream. The problem with this is it really is not able to account for the fact that domains probably are hierarchical. Just like I was saying, there's things going on at the sub-tad level, and you can see them here, right? So they call this a tad. But do you see triangles here, right? There's structure that is visible. And so there's a couple of methods. So this one is from Carl Kingsford's group. There's software available for all of these. So this takes in a heat map from one of these assays and actually tries to call a hierarchy of alternative domains. And so you have the original domains, and then you have subdomains. And so this is actually allowing you to start to resolve these structures. You've got to remember this is always resolution dependent. The more you sequence, the more you're going to see. And so the level of domains you call isn't necessarily the level that's there. You've got to calibrate the fact that how much sequencing you've done, also how many genome copies went in, and other things that affect the resolution. Another method that is also published and available, this is coming out in bioinformatics right now, but this is called TAD-tree. And it has a fairly satisfying idea. It uses something called the boundary index that we actually defined in that previous analysis, but what it tries to do is find a hierarchy of these TADs that is sort of optimizing the overall interaction frequency within a TAD, right? Because that's kind of what you want. You want to divide it up until within a given TAD, everything's about interacting at the same level, or at least that's a sensible way to try and do this. Okay, so those are two approaches you could use if you want to do TAD segmentation of these things. Okay, so back to some papers for a second. So we can do Hi-C. We can do various enrichment approaches to try and get better resolution, but it's worth asking how high a resolution can we achieve with Hi-C. And so this paper came out just under a year ago, a 3D map of human genome kilobase resolution from Rao and Lieberman Aiden's lab. And here they have single data sets with 4.9 billion mapped reads, so actual measured contacts in their deepest data set. The other thing that was done is this is where they introduce this in situ Hi-C protocol, where all of the cross-linking, digestion, and linking is done in the nucleus. And so this improves the resolution, improves the quality of these mappings. And the result is that, so this is the highest data set over here, and I'll zoom in a little. Yeah, you can see that now, where here, right, it looks like there's a domain here, but what can you say about what's going on? But now, with better resolution, better signal to noise, and better power, you can actually start to see much more, particularly these off-diagonal concentrations. This is really probably the best evidence now for the idea of actual looping in DNA, where you have two regions, here, this region and this region, that are coming together, that are very, very close, and the DNA in between them isn't even as close as those two regions. And so you can read that as one of these kind of classical loops that's been proposed for insulator, for insulators. So they developed this algorithm. This is a completely ad hoc algorithm for what it's worth, but it's smart. Basically what it does is it looks at a region, and it looks at various regions around it, and it's just looking for places where you are highly enriched relative to your surroundings. You need to do this because the actual level of interactions doesn't mean a ton globally, because it's very dependent on what the other structures are that you're contained in. If we think that this stuff is hierarchical, then a certain level of interaction here could be very strong, but here may not mean anything at all, depending on how compact the domain is contained in is. And so by just focusing on a relative kind of bump in these maps, they're able to call strong loop-like interactions. And so, yeah, down here I have they find about 10,000 of these in GM 12878, which is one of the tier 1 ENCODE cell lines, very, very well studied, and 3,000 in the mouse CH12 cell line. So that's not a huge number, right, of these loops genome-wide. They're definitely the strongest that you can possibly see in these cell types. So a couple of things to look at here. So if you're looking at percentage of peak loci bound, I don't actually remember what this fold change is about, but this direction is kind of important. You'll see there's certain transcription factors. So these are all transcription factors. And out here we have a few favorites. We have CTCF, we have RAD21-SC3 cohesion components, we have YY1. And so they're highly, highly enriched for these various factors. Now, what's very, very interesting, if you look in here, is actually that you can see at these sites where CTCF is binding that there's actually a directionality. And so this is, right, these are where these loops are occurring. And you see that the CTCF binding sites actually appear to have a particular orientation. If you look at all of these peaks now and look at the ones that have CTCF binding sites in pairs and ask, okay, how many of them are convergent, so facing each other, versus other orientations? 92% of these strong loops have CTCF sites at either end in this convergent orientation. Nuts. So nobody knows what to make of this, but it's a great idea. So let's go and actually edit some of these sites and see what happens. And so this is a paper, what's the date on this? I feel like it's like 6 months ago, so Ross already talked about CRISPR, good. So basically, they went into a particular gene cluster and asked what happens if we take these sites and we reverse them, okay? And so this is a mouse protocadherin gene cluster, and it's a bit interesting. So what you've got here are regulatory regions. So like this HS51 is the one they looked at here. These are CTCF binding sites, and there's 2 of them in this orientation. And so normally, what you have are interactions between this region and upstream cluster here. And similarly, there's an interaction here. And so what these are 4C profiles. So basically what that means is you take a single region and ask how much it's interacting with everything else. And so this means HS51 here is interacting a lot with stuff here. The higher the bar, the more it's interacting, and less with stuff here. And so now, if you go in and take this site and flip it around, so there's 2 CTCF binding sites, that's what CBS is standing for here. You flip it around, you see where before you had 72% of your interactions with this upstream region, it's now completely flipped. So by modifying 22 base pair sites, you've completely changed the chromatin architecture across 150 kilobases. And just for follow-up, they also looked at the beta globin locus and a particular domain downstream, upstream, away from, downstream.
The orientation, right? There's a number of things different ways. So the top line in blue, that's the nomenclature I'm familiar with.
Yeah.
Oh, that's the whole locus. Yeah, over there in blue.
Yeah, this is the
olfactory receptor.
Right. Right. So there at the TAD level, there's kind of 4 domains here. You can't see the CTCF sites? Oh, that's terrible. Well, there's CTCF sites at the ends of the domains, as one would expect. And so if you take, again, in the anchor site here, and reverse it, you see, so this is normal, right? So 13, 15 here is our anchor. We're looking at this domain. This is the normal case. And so everything interacting upstream. And you flip the site and you see this incredibly dramatic reversal of interactions between this region where previously it was strongly interacting to here. It's flipped around completely and is now strongly interacting the other way. Questions? Crazy, right?
Yeah.
All right. All right, so one more. This paper just came out, and for some reason it was unable to be downloaded from PNAS for like a week. And so I literally have only had a couple of days to look at it. But it's kind of interesting. So this is same group, Rao again, Lieberman Aiden, and this is the first author, Adrian Sanborn here. What they've done is they've done additional high C, substantial amounts of additional high C. They've also done some fish to confirm a number of these interactions and actually to inform their models. And there's some CRISPR in here that I'm actually not going to talk about, but it confirms some, maybe confirms some of the results. I haven't had enough time to study it. But one really interesting thing to me, at least, that came out of this is they looked at it just simply, okay, one of the things that happens in these experiments is you get a lot of these circle fragments, right? So sometimes you're going to get self-circularization of the DNA. So instead of getting a pair, you can get a circle. And so you can look at across different fragment lengths with different restriction enzymes, your frequency, your probability of getting cycles. And what they find is this peak at 1 kilobase, and actually a relative depletion out here at about 60, and so if from physical models, you would expect that if most of the chromatin were in the 30 nanometer fiber, you would see most of your circularization around here. But in fact, this is suggestive that a lot of the chromatin is more like a 10 nanometer fiber, and that's important because it means it can bend in much smaller amounts, right? So that means now instead of having DNA that can only form arcs that are around this 60 or half 60 kb length, you can form arcs that are 500 base pairs. Does that make sense? Cool. This is going to be hand wavy. I'm telling you this at the level I've actually read the paper. All right. So the next thing is, if you look, now that you have all this extra resolution, you can go back and redo that analysis that was done in the original Hi-C paper, which is that distribution we were just talking about, right? Look at the distance of interactions and their relative abundance in the actual observed data. And so what you see is that where previously they had this line that was about 1, that was because these short interactions were so poorly sampled because they didn't have any data there, right? And so if you go back, what you're actually seeing here is this negative 0.75 or negative 0.76 sort of slope on the contact probabilities. Good with that. So why is that a problem? Well, then what they do is they go back to their fractal model and they start making different kinds of fractal models. And basically through simulation, a whole theoretical thing, they show that there is no way that they can explain a slope of less than 1 using any of the fractal globule models. So the observed data requires this distribution with a lower slope than can be explained by their previously proposed model. So this is actually really nice because then they come up with an alternative, and that is, well, what if we say, okay, rather than, what if we basically increase this idea of internal forces? So what you're basically saying is that there are now things that are acting on the DNA at a short range that are putting pressure on it to bend and to form structures. And so if you increase that, which is kind of consistent with, we know we have proteins that do that, so if you increase that, and this is just showing if you keep increasing it and you do simulations, right? So you say, okay, I'm going to set up a physical model, I keep adding more and more of that internal strength, and eventually you can hit exactly that kind of 0.72 or so sort of model. And so this is the simulated, or 0.7, let's say. This is the simulated contact probability from a model that looks like this, where you have this internal pressure. And so this is just a cartoon, but showing under their model what the aggregation then looks like, right? And so you have your kind of spread out stuff, which looks not unlike the models that we reconstruct. And then as it's being compacted, it's compacting locally, and then finally you get kind of this structure. And so this model is very consistent with this slope. Again, it's still a model. It's still entirely based on one number, right? Fitting one number from the observed length distribution, but it has some appealing properties in terms of how we think this stuff works. And so here's a proposed model, putting this all together for how this crazy structure is working. And so somehow, these are two different approaches to this, but basically the idea is that somehow we get something that starts extruding the DNA. This is where, for this to be viable, you have to be able to bend the DNA, right, at a very small error. And so now if we can bend DNA at that level, then we can, for example, use cohesin rings and then that will slide along the DNA. Now in these two models, one is that the CTCF is actually pre-associated with the extruding complex here. In the other, it's actually already bound at its various binding sites. And then the idea is that it's sliding along, and when it reaches two sites that are in the right conformation, it's able to form this structure. And this is your insulator hub structure, right? This is what ends up forming the bases of the loops that we see. And alternatively, you could have this alternative model for explaining it. And so at the end of the day, then you end up forming these structures with these biased CTCF loops. And so you can ask all kinds of questions like, where does it start and why does it start there? And how does it skip over the CTCFs that are in here and things? But this is at least starting to look like some kind of sane model. I had another slide.
How does it skip over the CTCF?
No idea. I wish I could figure that out.
That's a huge problem.
It is a huge problem. I mean, it's not a huge problem. It's not a huge problem here, right? So if you make the argument that basically this whole thing's not going to stabilize until both of the CTCFs are binding a site in the right direction, right? Because CTCFs, what, it's got, how many zinc fingers does CTCF have now? A lot. And we know its conformation changes depending on what it's bound to, right? So we know that it will change binding outside of its core site depending on what this core site actually looks like. And so if anything was going to be able to bind different sites and stabilize its structure like this, maybe CTCF, you're going to, no? Yeah? You're the biochemist. I mean,
the slope for your power law come from the minus 0.75 come from a fine tuning of the tension parameter, or is it a very general thing that falls out?
No, it's totally right. So it requires a particular tension parameter. So this ratio R is your tension parameter, right? The ratio between internal and external force strength. So as you make more internal force strength, and so at an R of, or what? At a ratio of 10 to the 4, you're seeing that's where you get that 0.721.
But the slope is, the empirical data comes from many different cells, I would think.
Yes.
So something in many different cells is fine. I mean, if the model is correct, it requires that there be some tension parameter that's fine-tuned in the cell.
I don't know why you would say that. I mean, it's happening across many cells, but it's just a biophysical property of the combination of having organizing proteins and the pressure for a certain kind of conformation, right? It's a very global property.
Right. Well, I guess I'm contrasting it to, say, a model. So, for example, where parallels are seen in other contexts, you make some very general assumptions about, for example, how a transportation network will branch.
Mm-hmm.
The power law comes out of some very general assumptions about the structure of
Right.
the network. Here, it requires.
No. So you might be right, but I think not. You probably understand this better than I do, but it's always a power law, right? No matter what you assume this parameter is, it's always a power law. The question is just the particular slope that you get. They're just trying, and that the tuning definitely comes. The slope definitely comes from tuning. Yes, but the tuning is a global tuning to try and explain the slope aggregated across all those cells, and you see this, you see this at different lengths, and you'll see it in different. If you look at different experiments, you should see the same thing. But yeah, it's always an aggregate. That's the biggest problem with all these assays, right? Is for this to work, you have to go in with a couple 100 million genome copies. It's very cell expensive, and so it's always a very, very coarse ensemble of what's going on.
So back to the next slide. So were you saying, if you think about the extrusion happening in both directions simultaneously, then you're only going to get it to loop when the two CTCS hit
Yes.
and within some time window.
And that's why I asked, how does it know where to start? Because where it starts is actually, if it's happening in both at the same time, that's actually what determines the entire code, right? Mm-hmm. But they didn't answer that question. So we still have something to do.
But that's a helpful answer.
But I mean, again, it's a cartoon. I'm just trying to put some sense to it. There's no evidence for what I just said. I don't think they know, right? They don't know which direction it slides. There's lots of open questions, but at the same time, it explains it's a lot more than we had a couple months ago. So it's pretty interesting. So anyway, this kind of, if you put this together, you've got the TAD model. We can now envision a model for chromatin with loops of about a megabase down to maybe a couple hundred kilobases, formed essentially entirely by this CTCF code and then interactions between these, which is consistent with the old models we were talking about right at the beginning. Okay, so I'm going to finish with a little bit of practical recommendations. So we've built a lot of software to try and help people do this analysis. This is all in your notes. So we have a package called High5, and it's extensively documented and has tutorials. And basically, the idea here is we've made efficient data structures and file formats for working with this data. And so, for example, the Rao, a couple of billion fragments in their paper, they used a very, very large amount of computing power to analyze this. We can do it quite efficiently on just a small scale cluster or a little longer on an individual computer. There's lots of flexible tools for loading and filtering. You will find that your data set's going to be weird. There's going to be something odd about it. And so filtering is important. You have to look at this. We've implemented multiple normalization approaches. So we have a couple of different matrix balancing approaches, one that we've developed and a couple taken from other publications, and also probabilistic models. And these can be chained, so you can actually do one correction and combine it with another correction. The idea here is we think we have a good one, but the reality is not one approach is going to work for every data set. So we've tried to make it easy to compare different approaches and use the normalization that's going to work the best for you. And the way to evaluate this is actually, unfortunately, the best way we hardly have is looking at replicate correlation. And so you definitely want to have replicates, just like every experiment. And you can basically choose between normalizations by comparing replicates. And then there's various tools for binning and creating heat maps and visualizations and connecting with the various domain calling tools and such. Let's see, the other thing I do want to mention is upstream of this. I'll get to that at the end. Okay. So I'll just mention, like I said, we've evaluated different algorithms. The full probabilistic algorithm is definitely good, particularly for trans interactions, which I'm not showing here. But we have these quite efficient algorithms. This express algorithm is probably the best, but then we also have an implementation of, which is a similar approach that was used in that Rao paper if you wanted to try that as well. And so this works quite well, and these are dynamically binned heat maps that help reveal a lot of the structure. Performance-wise, again, minutes versus hundreds of minutes or hours, so this is fairly practical. We're also, this is kind of work in progress, but a lot of it's already up. We're basically processing and doing quality control reports for every published Hi-C data set, and so you can compare your data set to these other data sets, and you can see exactly the parameters we're using to normalize everything. And so we're hoping this is a useful resource for those of you who are trying to do your own Hi-C. And so we've got the scripts and commands to exactly reproduce this. This is on GitHub currently, and we'll be putting all the data out somewhere public soon. But everything needed to create the data, all the parameters and reports are there. I'll plug our little browser again. And finally, okay, what do you really want to do here? You're going to align your ends independently using a split mapping. We are highly recommending BWA MEM. This is a variant of BWA that works based on finding maximal exact matches. It's very good at dealing with the case where you have splits. So that would be like, we talked about splice read mapping, but in this case, we can also have chimeric reads. So what happens if you have read through, and so a read actually contains both ends of your Hi-C fragment. If you don't align those well, you're losing a ton of data. And so that, by using these split read mappers, and then, and the other thing is these have to be independent. So use the split read mapper, and then we can, we have tools to pair these up, resolving the chimeras. This will help you recover potentially a lot of missed reads. You must normalize. It's absolutely essential. You will see papers where they haven't normalized. You should ignore them, and you should normalize your data. You don't have to use our method. You can use many different people's methods, but you absolutely have to normalize. All the available domain calling approaches are pretty reasonable, and you should probably apply them all because it's very subjective. I can't yet make a call as to what the right method is. I do sort of like the TAD tree one that I mentioned at the end. I think visually and algorithmically it seems pretty intuitive. Calling point-to-point interactions is really, really dangerous because of this phenomena of everything being dependent on context, right? So it's knowing whether something is actually significant relative to the domain that it's in is very hard. So unless you have really, really, really rich data where you can see a strong point where it's surrounded by things that are not strong, I'd be very careful about that. That's why most analysis you see these days is still focusing at the level of domains. And then be even more careful about 3D models because these are all ensembles, and without fish data, they're really hard to calibrate. So you can do them and you can put them in your papers, but be careful about what you interpret from them because they don't reflect reality in any way. Okay, I hit my time again. Any questions? Yeah.
Yes, in the 5C, how do we know how to design the primers, not the university ones, the other ones? Is something by definition or?
Right. Yeah, so there's a couple of tools that are available for doing it. You basically will say, you say what restriction fragments you want to interrogate. And the tools are going to use sequence characteristics. They're going to use similarity to try and come up with optimal sets of primers. And so 5C primer is one. Those are actually quite good at what they do. And so it'll literally give you an ordering list for the set of restriction fragments you want to interrogate.
How many primers, more or less, do you need for these?
Need or want? Oh, well, it depends on what you're interrogating, right? So probably the best thing to look at is the Nora X chromosome 5C paper. I think the upper limit is in the several thousands. You can do several thousands if you need. I mean, basically, it's if you want 1 KB resolution, you need to use a 5 base per cutter, and then you need to use as many primers as the regions you want to interrogate. Exactly, right? Right. Makes sense.
So I guess I understand that downstream you really do want to look at human and mouse and really understand the chromatin structure on those levels, but has anyone taken a step back and maybe looked at a simpler system, maybe something with less DNA, and applied these principles to that?
Yes. So there is a yeast paper using a variant 4C technique, which is actually kind of like high C. So that shows some principles of chromatin organization. Now, yeast is different. Right? This is the problem, right? It's like, oh, let's look at a different chromatin context, and it's different, but it's different because it's organized differently. I mean, so there's one worm paper now. So worm doesn't have any of the, elegans doesn't have any of the canonical insulator proteins, and so it looks very different. But what that's telling us, I don't know. So the information is there. Those experiments are being done, but I'm not sure they're revealing that much right now.
Well, I guess that would be an interesting comparison to draw, is if you found similar structures in something you didn't expect to have insulators, maybe you wouldn't necessarily anymore attribute those to insulators. So I think
I agree.
that could be really useful.
So the question, despite all of this, the question of what's actually driving the organization of chromatin is still completely unanswered, right? So, insulators appear to be there in human, but they could be a side effect of a completely different process. So, I totally agree. So there's a strong argument that it's just a side effect of transcription, and I can't prove to you that that's wrong. I'm trying to. We're working on that one, but I can't. Based on all of the data that's out there, you can almost infer chromatin structure. Well, all of the.
You're saying that a lot of this chromosome conformation stuff could be a result of transcription, not leading to it. And if this is thin ice, but I'm going to use the word that some biochemists say, transcription factors.
Yep.
Right. And the reason I'm being indelicate about this is that there are two types of biochemists in the world: those who take transcription factors seriously, and those who absolutely hate them. And but I look at the pictures and I see all that bulk tube going to maybe 100 locations across the nucleus, and I said, looks like this all together. And so anyway, to be whatever you see as the nexus of interest in these high C patterns, it could be. That's where the polymerases are. That's where, and the answers.
And there's, at the boundaries, it's definitely clear. The signatures of transcription are there. You always find active genes at these boundaries. And so if I'm not taking a position on transcription factories, but if you told me the nucleus is just a checkerboard of transcription factories and polycomb bodies, I couldn't use this data to argue that you're wrong.
Those data
do not exclude that hypothesis.
Then they're consistent with the factory model, but they don't prove it either.
Yeah.
Isn't there some evidence from, I think it's from the software, that deleting CTCF doesn't delete chromosomes, it doesn't change chromosome organization very much.
Yeah.
So how do you fit that into this?
I don't know. Not only CTCF, but we knocked down CP190. We knocked out all 5. Couldn't detect anything.
Yeah.
So that's where I'm having a tough time.
So they may be stabilizing.
We are collaborators.
But that can't be the only thing that's going on, because you can get rid of it and you still have structure.
Yeah.
So I know there's also a lot of development in terms of
Mhm.
And so I wonder how that would, if you thought about how like advances in that technology would inform what you're doing.
You're thinking like imaging.
Yeah.
Yeah. So my favorite, there's been a bunch of great work in single cell, single molecule chromatin imaging and also high throughput. So that personally is where I'm hoping to go, because I think you can combine these models with high throughput imaging to actually get the stochastics onto these. Because I don't think they're wrong. They're just ensembles and they don't tell you the whole story. But if you have a few thousand observations of a few dozen loci in single cells, then that starts to become really interesting. And so, yeah, there's great work on actually real-time imaging of movement of chromatin, and
That's
very
what
exciting.
Are arguing with the people about transcription factories.
If you can also see what's going on at the same time, right? So most of these things are fantastically beautiful images of chromatin, but nothing else that's going on right now. But I mean, imaging is going fantastic right now. So imaging is the next genomics, right?
There is a new technique, it's called in situ sequencing. So once you can identify maybe two genes that are in close proximity by these approaches, maybe then in situ sequencing can be something good to visualize
Yeah.
these things in the nucleus.
Mm-hmm.
And regarding the CDCF, in this, I think that long non-coding RNAs can be mediating this starting
Yes.
point to bring CDCF.
Yeah, I mean, we know that in Drosophila, right? We know that at least gypsy has an associated long non-coding RNA, and there could be others. Well, no, as much as we know anything.
Have people used this technology to look at replicating cells or cells in meiosis where you're not having recombination events or you're having large changes in chromatin structure?
Probably. I don't have a categorical memory of all. I mean, that's something I think everybody wants to do, is look at this at high temporal resolution through every aspect of the cell cycle. It's not been done as much as you would think it would have been done, as far as I know. But certainly replication is interesting and has been studied a fair bit. And these, yes, one obvious thing is that these are totally, almost exactly associated with replication domains, right? So, but is that meaningful? I mean, everything is associated at that level.